Holy cow. Welcome to another edition of Mr. W Live. And you know who's here? It's Mr. W. And it's Mr. W with some of my favorite people, my people who have been showing up to all these Mr. W Live sessions. What a fantastic feeling that is. Who do we got? We got Jyoti, we got Matthew, Sophia, Emily, Flora, Carla, Matthew, just more people are showing up. Marion is here now. This is fantastic. I think some people might have been waiting for me. Very, very exciting to hear people. And I've got a special treat for you because I'm so excited about this thing that we're doing that I actually wrote a song about it. I've got my Mr. W Live theme song. And you guys are the first people who are ever going to hear it. And you know, I'm just going to do my best. And let's just check something here. Let's just do a music check. Let's do a little sound check, guys. Can you hear that okay? How'd that sound? Just somebody? Oh my God, I got someone from France. Holy cow, that is amazing. Bonjour, comment allez-vous? Je suis très heureux que j'ai des étudiants français qui vont à... that's about as far as I can do. But anyhow, all right. So guys, you guys hear that? Can I just hear one person just say, I got that sound. I heard that beautiful music. Okay, fantastic. Let's do it. Okay, you guys ready? Uh-oh. Got a lot of music playing for today. Listen to this. Mr. W's live on YouTube. Tuesdays and Thursdays, 2 p.m. PST. He's gonna help you learn AP biology. His BIOMANIA system will help you to review. He'll show you how to write your FRQs. Ask Mr. W all your questions. He's live. He'll guide you to a 4 or 5. Oh yeah. Guys, that was about the most fun I've had in maybe this entire pandemic. I am so stoked to be here teaching AP biology to people from all over the place. I've got Liv, I've got Carla. Holy cow. So this is fantastic. And now I'm hearing from Sputnik that they wrote très bien, because I spoke a little French. And you guys know I speak Spanish too. My Spanish is actually much better than my French. So si ustedes quieren un poquito de enseñanza de biología en español, yo puedo hacer eso también. Okay, guys, that's great. I'm hearing some good feedback. That's making me super happy. And how we doing here? Okay, so, well, we got music on the table, except, gee, I wonder where I put my lyrics. Oh my goodness. I'm not going to do that again. All right. Let's see, how can I make this work? I think I got a plan. Got a plan. It's very embarrassing when you're all ready. But this is what happens. I don't know if you guys know this. This happens to teachers all the time. They've got their lesson plans. They think it's perfect. And then they like put down some paper and they're not sure where they put it down. And then you might see them looking around like I am now. And then they're like frantically trying to do something else so that they can recover. It's just a difficult thing. So I think that what I'm going to do is, I think I know how to do this. I'm going to go here, and I'm going to go, you're really just seeing me do this, and here I am just sort of flailing around here at this moment, but you know, you guys are incredibly patient with me, and I really appreciate that. But I'm going to go here, and I'm going to go here. I'm bringing up a file is what I'm doing right here. You guys can't really see any of this magic, I'm hoping. But here we are, and here we are, and I think this is going to work. All right, I think I got something that is going to work. So here's the plan. Guys, what I want you to do is this, all right? So I want us to do some mitosis review. And the way I want us to do this mitosis review is to sing it. Now, I got to be honest, I don't think that the phases of mitosis are going to show up on this year's AP Bio test, right? But there might be some stuff related to cell division where it might be useful to understand these underlying concepts, and that's sort of how the content relates to the processes, and that relates to overall biology achievement. So let me just show you what I'm asking you to do here. So that's kind of a funny picture. So here's what you guys are going to do. You go to my website, right? So everybody's should be looking at my website right now. Let's see, I'm seeing what you're seeing. Fantastic. So if you click over here, click music videos, all right, and then click karaoke biology. Please don't ask me to speak Japanese because I can't do that. But if then you click mitosis karaoke, and then if you scroll down, you see in this PDF, you scroll a little bit more, there's the lyrics. And you know, I haven't set this up in the most convenient way, and I'm sorry about that. Do I? Oh, right below that, there's all the lyrics. That's golden. And in fact, I'm going to look at this too while I sing Mitosis. And what I'm hoping is that you guys sing with me, because this song has one of the greatest hooks of any of my songs. I don't think it goes on to the list of other great hooks in the sort of rap, rock and roll kind of genre. You know, this is just, in the world of biology songs, I think this is a great hook. But you know, that's a very narrow set of music. Okay, so let's hear some mitosis. Okay, so you guys are all going to be looking at this, okay? And maybe you'll also be having another screen where you'll look at those lyrics, because this is kind of difficult. These are blanks that are going to come in. So I hope you guys are ready to do some mitosis. And let's see what we got here. I brought this up for me so that I could follow this. Because, you know, just to be totally honest, sometimes I forget the lyrics to my own songs. And that's because that's the way that memory works. It's especially the way that memory works when you're 58 years old. But, you know, it's really, our memory is not like a computer memory. In other words, we form meaning associations, so we don't always get like the specific words in the right order, the right way. And that's sort of important, because that's why practicing for the AP Biology test in the way that I've been encouraging you to do it, it's almost kind of like you were practicing to deliver a speech in a history class or something like that. That is why that activity is so important, because that's how you develop the neural networks that lead to sophisticated thinking. And that's what you're going to need to do on May 18th, when those two questions come into your home. That's never happened before. You've gone to the questions, but now the questions are going to come to you, and you're going to have to be able to think biologically. And thinking biologically means understanding concepts, understanding processes, understanding data, and I'm going to talk about that in just a minute. So just so that people know that I'm not tracking questions right now, because I'm going to be focused on singing, and I'd like you to focus on singing too. Okay? You guys ready? Hope you are. Let me just see that the screen is looking good. Going to do a last check. Perfect. Perfect. Let's go. Let's do this. All right, so I'm going to go over here. Oops, not there. I'm going to go over here. I'm going to click. Guys, I want everyone's attention. We're going to go over what we did yesterday. We're going to go over this. Stages of mitosis. Sorry about that.
All right, everybody ready? All right, let's go.
Interphase of cell division's longest part. Nuclear membranes intact as it starts. The cell's growing, cytoplasm flowing, chromosomes get duplicated, DNA gets replicated, chromosomes are spread out so they can't be seen distinctly. But note the nucleolus, the ribosome factory outside the nucleus, the two centrosomes. They later make the spindle, which will pull apart the chromosomes. Prophase follows. The chromosomes condense. Each is made of two sister chromatids like an X. Each sister is a clone, the closest of kin, and a centromere connects them like Siamese twins. The nucleolus disappears, it melts away. As the cell takes a ribosome production holiday, the centrosome separates for spindle formation, for separating chromatids and cell elongation. Mitosis, chromosomal ride in a pro-meta-anatelophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew. Late prophase, pro-metaphase. The nuclear membrane disintegrates. The centrosomes migrate to the cell's opposing sides, and between them the fibers of the spindle wend and wide. The spindle is made of microtubule fibers which attach the chromosomes at kinetochores, a protein patch that serves like a handle that the fibers can grasp when they pull apart the chromosomes, splitting them in half. The spindle moves the chromosomes with nudges so fine into linear formation on the 50-yard line, a location equatorial defining metaphase where the chromosomes have lined up in that middle place. Mitosis, chromosomal ride in a pro-meta-anatelophase divide. Eukaryotes go from one cell to two. Mitosis, cells renew. The spindle fibers pull on the kinetochores. A cellular, molecular, mitotic tug of war. The centromere snaps, centers get separated. Now these chromatids are chromosomes. They've been upgraded. The snap and separation defines anaphase. The A for apartness, for moving different ways. Kinetochore spindle fibers separate the sisters. See them waving goodbye, calling out, I'm gonna miss you. And the other spindle fibers push and grapple like felons, make the cells elliptical like a watermelon. In telophase, membranes form around the chromosomes, which spread out as the nuclei come home. Mitosis, chromosomal ride in a pro-meta-anatelophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew. In animal cells, there's a ring of microfilaments that form at the equator, and they cinch themselves in tighter till the cell is in two pieces. Yeah, in animals, that's cytokinesis, but it's different in plants. And then the cell divides by building a new cell wall from the inside as the Golgi sends vesicles with cellulose goo, which makes a plate. Then a wall divides a cell in two. And instead of one mother cell, we now have daughters too. Identical twins, kind of old but kind of new. From your single cell beginning, this is how you grew. And for single cell eukaryotes, it's reproductive too. Mitosis, chromosomal ride in a pro-meta-anatelophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew. Oh, I can't do that. How cells renew. Woo! There it is.
Okay, guys. All right.
All right. That was fabulous. I got to take a little sip of something just to keep myself together. Wondering how everybody's doing here. Oh, there we are. There's the rap lyrics. And wow, I've got 18 people. And that actually makes me very happy. 18 people who are getting some good biology education. So I got a bunch of things. Let's see if there's anything in there. I think people are mostly chatting to each other, and that's beautiful. So we're forming a little AP Biology International community here. And here we are. All right. Guys, thank you. I'm seeing somebody say bravo and yay and fabulous, and I totally appreciate that. And again, there's the special thing that Mr. W asked you to do when we do these live events, which is the thing that's forbidden, the big sin in public education. And that is, please take out your phone and ask your friends to join us. All right? I'm going to be really happy. I'm happy with the 18 that are here, but let's say it were 180 or 1,800 or 18,000. Those are all good numbers. And it just makes me feel like I'm helping some of the folks who need some support, because there are 18 of you here. That's fantastic. But 250,000 kids are going to take the AP Bio test, and I just want to make sure that they've got a chance. So we've actually got a question about mitosis from none other than GOT. GOT, I've been noticing the chat, and I'm seeing how everybody really respects you for being right out there with questions. And let's look at your question. I have a question. So as anaphase occurs and the sister chromatids separate, the sister chromatids, yes, in fact, I have a line about that. Let's see if I can find it. The snapping separation defines anaphase. The A for apartness, for move in different ways. Kinetochore spindle fibers separate the sister, see him waving goodbye, calling out, I'm gonna miss ya. And the other spindle fibers, now that doesn't really say that. It's right up here. The centromere snaps, sisters get separated. Now these chromatids are chromosomes. They've been upgraded. So as long as those sisters are, gee, I can't really do that. They're connected like this, right, by the centromere. My knuckle is the centromere. Then, in that context, that makes them sister chromatids. But as soon as they separate, now they're chromosomes. I also want to apologize because I'm using this very, very old-fashioned term, Siamese twins. Now we call them conjoined twins. You know, the world is getting better. We're becoming much more sensitive to language, to different people's differences. And I want you to know that I am part of that world. I respect that. I get it. When I wrote that, I had a little lapse, and please forgive me. Please forgive me. Siamese twins, bad. Conjoined twins, good. If I ever make another version of the song, maybe I'll take that into account. Okay, so let's see. I answered Jyoti's question. Anything else? Can you please explain the Krebs cycle? I can't seem to understand it no matter how much I study, and I don't know why. Okay, listen. I'm not going to explain the Krebs cycle right now, but I do want to just show people things, all right? So here, if you're looking at my website, okay, so now there's my website. And how we doing here? I can go down a little bit. Nice, nice. So I'm going to click on AP College Bio, all right? So there I am, all right? And Matthew, I want you to follow this, okay, and everybody else too. So look through to Unit 3, Cellular Energetics, and then the Krebs cycle is a part of cellular respiration. So click into cellular respiration, and then click into the Krebs cycle. Now, Matthew, I don't want to put you on the spot. I don't want to put anybody on the spot, but the Krebs cycle is complex. Biology is complex. That's why I teach biology, because biology is so fantastic. Biology is complex, it's intricate, it's beautiful. So the main thing that I just want to emphasize is that you got to put the time in, but it's not a huge amount of time. You're going to go ahead, Matthew, and you're going to read this. And you know, I took the time to write all this stuff out, and then you're going to take all the quizzes. And you're just going to bust through all of these reactions. I break apart the whole thing for you. You're going to get down here, you're going to take this quiz, and then, you know, I've got a song. I've got a Krebs cycle song. So, excuse me, you can listen to those songs, and then you can bust through that too. But I assure you that by the time you get down here, you're going to get it. And you know, it might be an hour of your life, but that's going to be one of the most beautiful hours, because that's an hour that results in understanding biology. And there's nothing better than understanding biology. And Matthew, it might take some time. And GOT is right. GOT, you're the best. Thank you so much. Okay. Okay, and I got GOT's comments. And Matthew, I hope you're cool with that. And let's go on with what our lesson was for today. Okay, so we did this. Oh, got to tell you again, special deal. My iPhone app available right now for $6.99. Now the truth is that I told you that on Tuesday, and I forgot to set the price back. But normally it goes for $9.99, and it's $6.99 now. And you just go, and you got to download the app first of all, and then what you do is you do the in-app upgrade. And I just want to emphasize something, just so that everybody's really clear about this. It's not like I don't want to sell my product, but you know, here if you go to BIOMANIA, and then you go here, so like these are the same as the stuff that's on the phone, right? It's just in the phone, it's an incredibly convenient package. It's an incredibly convenient way to get this stuff. So you do have the same stuff on the website. And if you're in a teacher's class and your teacher is monitoring your progress on BIOMANIA, which I sure hope they are because it's a beautiful way to review, then the stuff that you do on the phone actually talks to our teacher tools, and you'll get credit. Like, for example, my students, I hope all of my students are using the phone, and you know, my students get a special deal because they're my students and I can't sell anything to my students. Okay, so good, Matthew, that's a very positive response to what I said. And someone just asked me per month. Oh, no, it's just one time forever. Toujours, Sputnik. It's, you buy it and it's you've got access to those questions forever. And Marion is talking about how many CO2 molecules are produced in 3 cycles of the Krebs cycle. Marion, here's how to think about this. I'm going to jump back to that. Why the heck not? Okay, so here we are. AP Bio. I'm navigating back to this. I'm going to go to the Krebs cycle. And here we are. It's in Unit 3, Cellular Respiration. And the Krebs cycle. And here's how to think about this. All right? Pyruvate. Pyruvate has 3 carbons in it. All right, it's a 3-carbon molecule. You take 6-carbon glucose, you break it down to 2 3-carbon molecules in glycolysis. Pyruvate in the cytoplasm goes through a channel in the mitochondrial membrane, and that takes you into the mitochondria, of course, into the matrix of the mitochondria. And the first thing that happens is what's called the link reaction. That breaks off one of the 3 carbons in pyruvate. It's a decarboxylation reaction, and that releases a CO2. You see that over here. Everybody go like this. Well, 1/3 of the CO2 that you just exhaled came from this reaction over here. Now, the remainder is a 2-carbon molecule, and that 2-carbon molecule gets attached to a kind of shuttle called acetyl-CoA, and that acetyl-CoA brings it to the reactions of the Krebs cycle. Now, it's not really a physical bring because like everything is happening in the matrix, and this is just a cyclical series of reactions. But during the Krebs cycle, the 2 carbons that are carried in to acetyl-CoA, 2 carbons that are full of energy, by the way, those also get decarboxylated in the reactions of the Krebs cycle. Let's see if we can find that somewhere here. Yes. So here's the 2-carbon acetyl-CoA that goes in, and then over here you can see one of those is getting removed, and here's another one that's getting removed. And you know what else is happening? What else is happening is that the energy, the chemical energy between these bonds, particularly between the carbon and these hydrogens over here, is being used to reduce NAD plus to NADH over here, and then reduce FAD to FADH2. And those molecules are what power the electron transport chain in the inner mitochondrial membrane. And that is a kind of go-to thing because that is physically localized in that space, okay? So, Marion, I don't really know if I could do that. And let's see, what else? Jyoti is answering some questions. Fantastic. All right. Marion, I hope that answered your question. But, you know, that's enough of the Krebs cycle for right now. Krebs cycle, fantastic thing. And I gave my little plug for my iPhone app, and I hope to do that again later. So, a couple of people ended last session with a couple of questions. And the questions that you asked were fantastic questions. They were about really tough stuff. And one of them is a ridiculously hard question. Like, I think it sort of goes beyond the bounds of the course. And in fact, I'm nervous. I'm nervous about this question because I think it's too hard. I'm just going to jump in, and I'm noticing that Carla is saying that she doesn't really understand pH and its importance in general. Yeah, Carla, let me just say this about pH. pH is acidity or alkalinity, all right? pH of 1, totally acidic. pH of 14, totally basic or alkaline. Where is this important? It's the difference that tends to be important. The main way that I think it shows up, well, it shows up in a bunch of places, but living things are constantly pumping protons around. And when you pump protons into an enclosed compartment, like for example the inner mitochondrial membrane, let me say that again. When you pump protons into a closed compartment, so like for example, the thylakoid space in photosynthesis or the intermembrane space, you're pumping in protons, that's lowering the pH of that region. And that just means that there's a ton of protons that are trapped in that confined space. And that creates a diffusion gradient, and those protons can move back in the other direction. They move back through diffusion, and that diffusion, that kinetic energy of diffusion, is what powers ATP synthesis as these molecules diffuse through the ATP synthase channel. So let me just tell you a couple of things. Carla, I'm just going to jump back here for a second. And first of all, two things. Oops. So Carla, number one, AP College Bio. Okay, and I want you to know that if you go to basic structure of water and hydrogen bonding, okay, so over here I have a little tutorial about acids, bases, and the pH scale. And you know, biology ain't chemistry. And if you know the stuff in here, that's sort of more than enough for you to know that, like later in the course, as we're talking about acidity and alkalinity and stuff like that, this will do it for you. This is going to carry you through. It's going to teach you about the pH scale, a little bit about buffers. Like, seriously, you don't need to know much more than that. So, Carla, that's for you. That's very, very good, Emily. Always right there. Fantastic. Talking about how cells, living things, do a tremendous amount of work to maintain acidity, alkalinity within certain narrow ranges. So like, for example, your pH is your stomach has a highly acidic pH. It takes a lot of energy to create that, pumping a lot of hydrochloric acid into that enclosed chamber. Cells, in general, tend to require a very narrow range of pH. So living things do all these kinds of things in order to maintain the acidity and alkalinity. In fact, there are conditions, medical conditions like acidosis, which is when the blood becomes too acidic, or alkalosis, which is when the blood becomes too alkaline. And again, a big part of homeostasis is maintaining pH balance, but for better or for worse, that's not a huge part of the AP Bio curriculum. And the reason why it's not such a big part of the AP Bio curriculum is that we don't really have the kidney isn't a big part of it. It's a little part of it. You know, I'd suggest you know about osmoregulation, and I've got a tutorial about that. And N is asking me to do a live stream where I work through College Board FRQs in real time. And here's what's kind of sad about that. I actually can't. And I've actually contacted the folks at the College Board to ask for permission for that. And that stuff it's College Board material. And, you know, my website is monetized, and I'm a business. And so they're not giving me access to that material. So I can do my FRQs, and that's about what I can do. I actually talk about how to master those FRQs in some other videos, and it's really about deeply studying the scoring guides and really working through those. And sometimes you have to read really slowly, and you got to read multiple times, and you're going to say, what's going on here? But that is my suggestion to you. And so many FRQs are online at the College Board's website. And I can actually show you that right now. So for example, if you just type in AP AP. I should be able to type that. Bio FRQs. You'll go to here. And you've got the questions for each year, and you've got the scoring guidelines, and you've got sample answers. And I would just say study those like they were holy writ. If you're religious and that speaks to you, study them like they're important to you. Study them like they're the Constitution of the United States or the Declaration of Independence or the Gettysburg Address or something like that. And that's really the main thing that I can offer. I mean, for my students, I can do stuff a little bit differently. But, for you guys, this is part of a slightly commercial endeavor. It's only slightly because that's how much money I'm making from it. I wish it were a major commercial endeavor, but that's besides the point. But anyhow, and I am sorry. That's all that I can do right now relating to that. So what we were going to do is we were going to go over some questions from the last session. And wow, this is a lot of text on the screen. So what I'm going to do is this. I'm going to just go over here. I'm going to put myself on this side of the screen. And I'm just going to read this aloud. That sounds incredibly boring. Why don't you guys just go ahead and take a minute and read this question. It showed up as a question that one of you asked me in the BIOMANIA app. So go ahead and read this. I'm just sort of going to read it silently, and that'll help me time how long it'll take you to read it, and you'll see me reading at my other screen. Okay, so I just finished the first paragraph. Now we're reading about Alyssa and her brother. Okay, go ahead and read the prompts for part 1, 2, 3, and 4. Go ahead and do that, please. Okay, I'm just going to be totally honest. I spent about an hour working through this question, and I don't want to diminish my teaching authority, but I'm still pretty shaky on it. It's that difficult. But here's just a couple things to understand. So a marker is a piece of DNA that you can attach a probe to that's close to a gene of interest. It's not the gene. It's just close to it. It's a marker. So historically, the reason why this is important is that, for example, when they were tracking down the gene for Huntington's disease, that's an autosomal dominant condition. It affected Woody Guthrie, among others. Is that right? Yes. And, Woody Guthrie, this land is your land. I hope I have that right. Anyhow, I'm thinking that that's not completely right, but I'll check it. So in the meanwhile, markers are close to genes. Huntington's disease is one disease for which they were able to find a marker long before they were able to find the actual disease. And that marker helps you track inheritance. And here's the deal. We're trying to find how the markers relate to the actual gene, and that's what the parts are. So I'm just going to show you a couple of diagrams that I drew to kind of help us through this, okay? The easiest one is Alyssa's dad. Now here's the deal. One thing that you're bringing to this question is the idea that hemophilia is caused by a gene on the X chromosome. Women, females, have 2 X chromosomes, and males have only 1. So, dad, we know that he's XB, he has that marker, and we also know that he doesn't have hemophilia. And yet this is just part of the critical reading that's essential to the AP exam. DNA testing has uncovered the following markers, and it also says neither Alyssa nor anyone else in her family has this condition. So dad is XB. We know he has the marker, and we're going to put a plus sign on this same chromosome. This is a linked allele, and that's because the plus is the wild type. He does not have hemophilia. Now, Alyssa's brother, we don't know what marker he has. I don't know why. Maybe he passed away before the test was able to be administered or something like that, or he just wasn't around when the doctor came by to get the marker. But he could be either XA or XB, but we know that he has the mutant allele over here. Okay, now, the mom. The mom, we know she's XA, XB. We know that in a sex-linked condition like hemophilia, the mom is the one who passes the condition to the offspring. Okay, it's not the dad. The mom, even though it's inherited primarily by males, it's not the dad who passes it on, it's the mom. So the brother over here inherited one of these markers and the mutated allele from the mom, and we actually know very little about her. So that's kind of what we know from the setup to the problem, just by doing some preliminary analysis. Now, I'll show you the answer. Alyssa's chromosomes, there are two possibilities. One is she is heterozygous for this, and then homozygous over here. All right, oops. And let's just read the explanation over here. Alyssa's father was not a hemophiliac. His marker is XB. Therefore, one of Alyssa's chromosomes, the one she inherits from her father, has to be XB plus. And that plus indicates the normal allele, the wild type allele. So Alyssa has to have this XB plus over here that she inherited from her dad. And also, in case you're thinking like, well, what if there was crossing over? That doesn't happen with males passing on their X chromosome because the X has nothing to cross over with, all right? So we know therefore that Alyssa inherited, now, all right, so now it's stated in the question, in the data table for the question that Alyssa is a heterozygote for the marker XAXB. That's why she's got to be XAXB. We know, therefore, that Alyssa inherited her XA marker from her mother, right? Because Alyssa's mother was a carrier for hemophilia, she could have passed on either of her X chromosomes to Alyssa. The two possibilities on this chromosome are X plus with the normal allele or XM over here. So from the dad, and then two possibilities from the mom. Now, because the mom's a carrier, there's, I believe, a 50% probability that Alyssa would be either this or this. And then, if she is a carrier, she'd have to be this over here. So I hope that was hopeful. I know that question came up yesterday. And Jyoti is saying, so the mom must be a carrier? Absolutely, mom had to be a carrier. And the reason why mom had to be a carrier is that if an unaffected mom passes on a sex-linked allele to her son, the only way the son can get it is from the mom, right? Because dad passes on the Y chromosome to the son. That means the X chromosome that the male child inherits has got to be from the mom. So mom indeed must be a carrier. Carla, what are linked alleles? This is a great example of linked alleles. Linked alleles are alleles that are on the same chromosome. They're physically linked. They're on the same piece of DNA. Genes, alleles, are pieces of information that are on, that are chemically encoded. So linked alleles, like in the same way as like this knuckle is linked to this knuckle, those are linked joints, all right? So if these were genes, they'd be linked genes. Genes on the same chromosome, those are linked genes. And Matthew, Jyoti, well, fantastic. I achieved my goal. That made some sense. So the other question that came up from last week is this one. It was the chi-square question. Now, let me say something. I don't want to make predictions about this year's exam, but I don't think it's likely that you're going to have to work a chi-square problem. And the reason why I think that is because it takes a lot of time to do those problems, you know. I mean, I guess you could have it programmed into your calculator or something like that, but these take a lot of time. I'm going to, by the way, move myself over to here, secondary screen. Where am I? Here. Yes, there I am again. So I'm going to walk you through this. And one of the reasons why I want to walk you through this is just because I want you to know that chi-squared doesn't only come up in the context of genetics. So here you've got these bees, and they do this thing called nursing behavior. And in the nursing behavior, what they do is they take care of the little larvae that are inside their little cocoons that are about to, you know, come out and be worker bees. So they're measuring the behavior of these bees in terms of whether they're night owls or early risers or somewhere in between. So these are the guys who are in between. These are the guys who are active all the time during the day. And these are the guys who are the night owls. And what you'd expect in a population is that there'd be a normal distribution of almost any complex trait. In other words, most individuals are going to be somewhat average, but then there are going to be some over on each extreme. That's a very common thing. You know, if you look at height in a population, that's really how it works. If you looked at sort of like how far people can throw a ball, probably falls down this way. If you looked at a large population and you were measuring like test performance on the AP exam, well, that's what you'd see too. You know, a lot of these things have a normal curve. There are teachers who grade on a normal curve, and I think that's a bad idea. I don't do that, but that's what some teachers do. Now, here's the deal. They're expecting a normal distribution, but then when they measure it, they find that, okay, there's a lot that are over here, and there's a few that are over here in the extremes, but then this bar is sort of way out of whack. So here's the deal. I don't want to go through all the details of this, but the null hypothesis is that your data is going to accord with your expectation, and that the difference between what you observe and what you measure is just due to some random, unimportant fluctuation. That's the null hypothesis, that there's just random fluctuation between observed results and expected results. So what we expect is in the black bar, and what we're observing is in the white bar. But how do we know? Because, like, for example, if you're flipping a coin and you got 45 heads and 55 tails and 100 flips, you probably would not be upset. But if you got 30 and 70, you'd probably start to think of it as a trick coin. And the chi-square tests enable you to evaluate this idea. So here's the chi-square formula. I'm not going to go through it, but I do have a tutorial. In my genetics unit, I have a tutorial that very sequentially walks you through chi-square. And if you feel that it's important and you feel that that's something that you want to master, then by all means, go ahead and do that. So this is called a critical values table, and it lets you look up what you calculate from chi-square. What you wind up calculating is you wind up calculating this value, all right, through this formula, and then you look up what you got over here. So you're evaluating your data range from 30, 40, 50, 60, 70. You're evaluating these numbers against your expected values. Let me just show you the next thing. So what you find, and this is actually, just to let you know, it's hard to do this. I did it in a spreadsheet. I actually grabbed this spreadsheet over here. When I ran my chi-square, my total was 7.47. Now, what I measured here was 1, 2, 3, 4, 5 things. And when you use this critical values table, what you do is you compare your chi-square value to a critical value based on your degrees of freedom. And man, this is complex, which is why I want you to go through the tutorial. And I just want to say in general, learning from videos, it's great. You know, there's so many great video instructors on the internet who are teaching biology. But you should be working problems. That's a much more effective way for you to learn. And that's why I'm really encouraging people to use BIOMANIA to do problem solving, because that's how you're going to get yourself to a really high level. So anyhow, degrees of freedom -1, we've got 1, 2, 3, 4, 5 categories. Our degrees of freedom is 4, all right? So we're going to look up our critical value at 747. We're comparing it to this over here, 949. 747 is less than 949. And what that means is that we can accept our null hypothesis. So, Jyoti, I'm going to go into your second question. So we assume that the observed values match the expected values and that the treatment has no effect on the results in a null hypothesis. I wouldn't say no results, no effect. The null hypothesis is that the difference between the treatment and your expectation is not going to be statistically significant. And in fact, the whole point is that there won't be no difference. There's going to be some difference, but chi-square tells you whether to worry about it or not. So like, for example, in this case, if this value had been more than 9.49, then you worry about it and you think, oh, the bees biology is one where they're being encouraged to do more nighttime activity, all right? But because it's less than this value, we're saying just random fluctuation. It's as if I tossed a coin and I got 48.52 and it's not going to bother me. And Carla, how do we know which one to use? Okay, use 0.05. I actually can't explain why, because that's sort of like science culture. In general, scientists use the 0.05 value probability to decide. So that's all you need to do in AP Bio. 0.05. Don't take it any further than that. You're never going to do 0.01. You're never going to do 0.001. And if you start worrying about it, worry about that after May 18th. Get a book on statistics and read it. Don't worry about that now. Just know 0.05 and how to use degrees of freedom. And again, I'm not going to bet on what's going to be on the test and what won't be on the test, but I think it's, as you guys would say, hella unlikely that there's going to be a chi-square problem. I just think that. Jody's talking about, I think that's on the 2017 test. I just went over that with my students. And again, what Jody is saying is again, totally encouraging me to tell you something, which is this. Study these so intensely. You should be able to sort of recite the answers. Talk about them to your parents, your brother, your sister, the kids you're babysitting for. It doesn't matter. You should be able to talk about these with tremendous familiarity and fluency. There's actually additional ones that the College Board has put up for you on AP Classroom. You go into your AP Classroom and then they say like optional FRQs, so you got some more over there. And your teacher can give you additional ones from AP Classroom that they have access to. So there we go. Okay, so someone who's called Oh Yeah is saying that they listen to my cell song and that helps. And what could make me happier than that? So let's do this. Let's see what else I had planned today. I'm noticing that it's already 2:51. I'm going to play this just because it makes me so happy when I play it. I'm going to do it right now. I can't resist. You guys ready? You guys ready?
AP Bio Review. FRQ.
Sometimes when I'm feeling a little blue because of the pandemic, I just play that song and it just makes me happy. Just makes me happy. And again, that's the brilliant Max Cowan who took my voice and made it sound that good. That is really something. Guys, let's do one problem. I might go over an hour. I mean, anybody have anywhere to go? I don't really have anywhere to go. Actually, I have to go to the hardware store. I gotta pick up some stuff for our garden. It's kind of cool. Want to check something. Guys, has everybody texted a friend and said to that friend, hey, Mr. W's live. I'd like you to join us. But in the meanwhile, let's do this. I got a genetics problem for you, okay? And it's a two-part genetics problem. So I want you to go ahead and piece of scratch pad or something like that, and I'm going to answer. Carla, I don't know what's going on with AP Classroom. I found it incredibly difficult, and I'm just sort of sharing stuff with it to my students, and I sort of kind of flaked out on that. Yeah, GRT, I got to think about that because I'm sort of remembering the question, but not well enough to really respond. And Emily, are the optional practice FRQs more likely to be accurate to the test as opposed? I would check out those optional FRQs. I'm going to send an email to you and everybody else about that. Yeah, do that. I'm also going to tell everybody one other thing before we stop. But anyhow, guys, work this problem. Do it now, okay? So everybody's going to work this problem, and I'm just going to sit here and give you time to work this problem. That's kind of, I should get some background music to play. I should get some background. In fact, maybe I'm going to try and do that. I don't know if I can pull that off. I'm going to try and grab some background music. You guys just work on this problem. Oh, fantastic. Keep working. Everybody keep working. Everybody's working these problems. Okay. I'm already seeing some answers coming in, and that's telling me that it's time for me to stop flailing around here and looking for music files that I can play in the background. And in fact, let's go. Okay, so we've got these parakeets, green feathers, big G dominant to little g, yellow beak, big Y dominant to black beak, little y. We got big G, little Y, little y is mated with a parakeet, little g, y, y. Now, just want to say, it's kind of worthwhile to memorize this kind of cross. And that's because this is what we call a test cross. We're mating with a recessive over here. And so if you have a double heterozygote, big G, big Y, little y, the result is indeed 1 to 1 to 1 to 1. And here's how you would do it, okay? So you know that like your Punnett squares, they don't have to be 4 by 4. They just need to be as big as they need to be. So in this case, you can only produce one kind of gamete, little g, little y. That's what you'd put over here. And then here you have to use the FOIL method. So first, big G, big Y, outside, FO, big G, little y, inside, little g, big Y, little g, little y. Now again, any problems with this? I have a tutorial on dihybrid crosses. And go do that tutorial if you're having any difficulty. So it's a 1 to 1 to 1 ratio, right? That's what you're expecting. Now, you count them. You count up all the offspring. Now, this wouldn't be just from one parakeet because they wouldn't have that many offspring. After all, they're not fruit flies or anything like that. But these are parakeets. You're doing some kind of population study, and you're observing the number of offspring, and you're noticing that they're not 1 to 1 to 1 to 1. In fact, they're like, there's one class, green, yellow, blue, black, that there are a lot of, and there's just a little bit. So here's my challenge to you. It's a difficult challenge. In one word, actually 2 words, blank and blank. What's going on? What's happening? What's happening in this cross? Why are the ratios so different from what you'd expect from this 1 to 1 to 1 to 1 ratio. All right? Can anybody put that in the chat right now? And this is why I need some music to make this happen. Anybody put this in the chat? Anybody? Got anybody who's going to do this? I'm waiting. And I got one answer. Okay, so guys, what you got going on here is this. These genes are linked, all right? And in fact, you can tell because you've got two classes that are just like the parents, all right? So green, parakeet with genotype GGYY, right? So that means it's going to be green feathers and yellow beak. Green feathers, yellow beak. And then the other parent is going to have blue feathers and a black beak over here. And notice that those are the parental phenotypes. There are always going to be more of the parental phenotypes, and then these are explained by crossing over. Okay? So guys, I actually kind of like this because, we dealt with some of your questions. And, we're at 2:58. Let's see what we're going to do now. Oh, little reminder that we've got the $6.99 deal for BIOMANIA. How about this? Oh, Marion, are you in the cis position or the trans position? Cis would be the same, like on the same chromosome, the same one of the homologous pairs, and then the trans would be the other. I don't actually know that. I wouldn't jump to use that shorthand. I'd actually say what it is. So guys, here's what I got. I've got paper. It's a blank sheet of paper. And if people have questions that they want me to bring to the next one of these sessions, which is planned for Tuesday, what are some of your questions? Anybody want to put a question in, or just put a topic and I'll go over that topic. So anybody want to put a topic in, and I will go to here. So I'm waiting for you to put in the chat some topics that you'd be interested in me addressing on Tuesday. And anybody got anything? So here, I'm just going to put what topics would you like me to address next week? Conceptual analysis. All right, we got gene regulation. Conceptual analysis. Jody, what do you mean by conceptual analysis? Eukaryotic gene regulation. I can talk a little bit about that. Genetics in general, viruses. All right, you know what I'll do? I will work up okay, so Emily, BIOMANIA FRQ. It's about apoptosis and mutation on different enzymes. Okay. All right. So here's what we're going to do. We're going to stop in a minute, but I want to show you guys a couple of things. All right. So let's jump back to my website. Okay. And here's one thing I want you to notice. So first of all, I want you to go to Science Music Videos after, when we're done. And you're not looking at that. I got to go like this. There we are. Okay. And I want you to hit live question of the day. And then what I want you to do is, okay, so hit question of the day right over here. And then you see this? This is a video that I just released yesterday on YouTube, and it's about experimental design. Now, we got some pretty sharp folks on the other end of this screen. And it might be elementary for some of you, but I do talk about like some of the stuff that I talked about on Tuesday. In terms of how, about experimental group, control group, dependent variable, independent variable, all that stuff. And it talks about data analysis, it talks about error bars, things like that. So I totally want to encourage you to view this video. And yeah, so I'm actually thinking of putting together another video about question 4. And so I'll work something up. And maybe that means, that might get in the way of gene regulation or not. So, and Emily, you're telling me about the one about TNF. Yes, BC12. I know exactly which one you're talking about, and caspases. Okay, so my promise to you is I will talk about question 4. I'll bring in that question specifically. And here's what you guys can do. Along the way, if you're using the BIOMANIA app or on the website, and like I sure hope you are, because however much you're enjoying these, that is a million times better. Like for example, for you to be spending your time not watching videos, but doing this, sort of working your way through FRQ after FRQ after FRQ, that is the way to get to mastery. Like there's just no other way. We can't, there's no way to short circuit it. There's no way to make it easier. You just have to do it that way. And I'm a little worried because I'm seeing a little problem on my website, but I'll fix it later. You have a website, it's just there we go. Okay. So just work these, spend time working this. Go like this, choose 1 through 6, and spend time. And again, it's $19 for a subscription to my website if you're not in a class, and it's the best investment that you can make in learning biology and getting yourself ready for the AP exam. And GOT, that is exactly right. We're talking about disruption to a biological system. And so that's what we're going to do on Friday. And, on Friday. What did I mean by that? I meant on Tuesday. And we are not going to do these quick questions, so heck, that would have been fun. But I do want to say a couple of things to you, a couple of moments of inspiration. Here, guys, you can do this. You can totally do this. Just work with BIOMANIA, work with lots of questions. Do that. That's the thing. Question yourself over and over again, work through really deeply understanding, and you're going to set yourself up to get to your goal. And I know that your goal is getting up here to a 4 or a 5. Keep practicing. College board questions, my website, the BIOMANIA app. Right now, run over to your folks and say, please, can I buy it? It's $5.99, $6.99. And that's it. And I'll see you here again on Tuesday. It's going to be super fun. Have a great weekend. You guys are awesome. I'm going to sign out. I want you to all take care of yourselves. Be well. Stay safe. Practice social distancing. Whatever state you live in, practice social distancing, because those states that are moving to end social distancing, they're doing it too soon. What we need to have in effect is something called contact tracing and testing. And we don't have that yet. Not enough. So anyhow, guys, with those words, take it easy. Study a lot of biology over the weekend. I'll see you on Tuesday. You guys are the best. Looking forward to seeing you again. Mr. W is signing out. Take care.